Hello, my name is Florian Duchatel. I'm Mark Bronsvoort's PhD student. So today I will present a novel modeling approach for endemic FMD in sub-Saharan Africa. So as you probably know, to be able to control foot and mouth disease, we need a good understanding of the epidemiology of the disease. And what I'm interested in today, it's to study the transmission rates between locations where we can detect FMD, the number of transitions between those locations, and the effect of anthropological and environmental variables on the virus transmission. So in sub-Saharan Africa, we've got quite a huge overlap of serotype and topotype FMD viruses, with some serotype from O, A, SAT1, and SAT2. And we've got apparently a great circulation between the different countries in sub-Saharan Africa. But the epidemiology of FMD in Africa, it's still unclear. We still don't know exactly the role of livestock and wildlife in the virus transmission. And the main problem too is that we've got quite a lack of data that make us less able to understand properly the transmission and the epidemiology of the virus. What I'm interested in is in the Bayesian phylogeographic reconstruction of the virus, which makes us able to incorporate spatial and temporal information in the virus transmission. As you probably all know, is that viruses are evolving so fast that within months or years, we're able to detect genetic differences between two strains. And those genetic differences, genetic distance increase with the time. That make us able to imply the existence of a molecular clock. We just express the link between evolutionary rates and time. Once we know, like, we think that molecular clocks exist, we can create time-scaled evolutionary trees where the branch length here are in years and express the time of divergence between the two branches here. We can also incorporate spatial data in those trees, for example, locations that we will use as evolutionary trees. And from the data, we can estimate transmission rates between the different locations we've incorporated. For example, here from location A, we've got transmission to a location B, C, and D. And those transmission rates are expressed as a matrix of transmission. For example, here, it expresses the transmission from the location A to the location B, A to C, A to D. But all those transmission rates are not, they do not have the same probability to exist. So we have to determine the well-supported rate of transmission between locations. To do so, we switch those transmission rates on and off to determine which ones have the highest importance in the phylogeny reconstruction. And at the end, we are able to test which one are the well-supported rates of transmissions between locations. We are also able to count the number of location transitions among the trees. For example, here, from the same location A, we've got a transmission to a location B, a location C, and a location D. We use those different techniques on two different data sets, one of FMD-A and another one of FMD-SAT2 in different countries, here like Kenya, Tanzania, and here like a bit the same. And what we've been able is to reconstruct the evolutionary tree, to determine the well-supported route of transmission, and to count the number of jumps between locations. What you can see here is that for FMD-A, we've got a clear separation between sequences from the west coast and those from the east coast. West coast are comprising Cameroon, Benin, Nigeria, and Niger, when the east coast are comprising Uganda, Ethiopia, and Kenya, and so on. we can also see that like the most recent common ancestor between all those different sequences was in 1920, which is quite recent. We've been able to determine the well-supported route of transmission, and we can also there see the clear separations between the two coasts with no exact, because we've not been able to detect a well-supported rate of transmission between the those two parts of Africa. We've been able to see some jumps from Kenya to Tanzania, Ethiopia, Uganda, and from Ethiopia to Kenya, but none toward like for the western coast, for example. But we've been able to see some from Cameroon to Nigeria and Togo. We did the same for SAT2, of course, and one of the first things we can see is that the most common ancestor between all the samples we have is way older, around 1300s. We've been able also to detect some well-supported route of transmission, for example, from Kenya to Tanzania, Kenya to Zimbabwe, and some jumps again, from Kenya to Tanzania, Zimbabwe, and from Zimbabwe to Kenya. The second things we tried to test is to understand the effect of environmental and anthropological factors on the transmission of the virus. Therefore, we used what we call a generalized linear model. So each one of the transmission rates between the locations is expressed as a linear function between all the different variables. For example, for the rate here from A to D, it's a function of the predictor, for example, human density, cattle density, or like different landscape density, such as crop forests. So we've been able to test the effect of different factors on the transmission, and the result is for FMDA, we've been able to detect a huge effect of human density on the viral transmission, where for FMD-SAT2, we haven't been able to detect such a strong effect as human density, but we've been able to see some effect of cattle density and distance on the virus transmission, expressing that like between the two different serotypes, there is a clear difference in viral transmission cause. So, in conclusion, what we did here is like we saw that there was a clear separation between the two African coasts in recent circulation for FMD-SAT1. We detected some well-supported route of transmission, and we've been able to see the effect of human density on the transmission of the virus for this serotype. For FMD-SAT2, we can see that the virus is circulating in Africa for way longer than FMD-A. We've been also be able to detect some well-supported route of transmission, and we've been able to see some small effect of distance and cattle density on the transmission of the virus. So yeah, those are the principal conclusions we've got so far. But one thing we really need, and that will make us able to have like better results, or not better, but like more strong results, is more FMD sequences with strong spatial information, because we need those ones to be able to detect environmental factors or anthropological factors in the transmission of the virus. And that's all. Thank you for your attention. So I will say thank you very much to all the other persons who participated in this project. Those from the Roslin Institute, with my two supervisors and my co-PhD students, people from the Institute of Evolutionary Biology at the University of Edinburgh, people from the Pilgrim Institute, from the Cameroon Academy of Science, the School of Veterinary Medicine and Science from Yaoundé, and the Institute of Aging and Chronic Disease from Liverpool. Thank you very much.
Florian, thank you so much. Sorry, I didn't know that you're going to give the talk, but thank you for very interesting. So I think we have some time now, and